pleasure to introduce our next speaker, Dr. Fran Sverdrup from Department of Biochemistry and Molecular Biology at St. Louis University. His lab specializes in early phases of drug discovery, includes target identification, validation, drug screening, identifying the mechanism of actions, preclinical evaluation. He's been involved with quite a bit of research, and his current area of focus is epigenetic control and gene expression in FSHD. Without further ado, Fran, the screen is yours.
Thank you, Jamshid. So I'm going to talk about small molecule drug discovery, and for the context of this presentation, small molecule drug refers to a drug that can be taken in a pill form. The pill is easy to take, generally the cheapest to make, and is therefore highly preferable if it can be done. But today I'm going to talk about the challenges for that to happen, which are significant. So I'm going to go over a few things that delve into the science and the biology of FSHD again, because there's no getting around that these challenges are sort of deeply rooted in this complex biology. So I'll touch on the central dogma of biology and gene activation, or what turning on a gene means. DNA, again, as Eric had mentioned, in chromatin, in the closed condensed versus open decondensed state. Open being sort of a hallmark of FSHD. What was noticed, one of the first things is that the DNA around D4Z4 repeats becomes decondensed, at least partway. And then once you have open or decondensed chromatin, how do you actually turn on DUX4? I'll touch on the cell and animal models that we need to test drugs, and an example of a p38 inhibitor turning off DUX4 in FSHD muscle cells and in an animal model. So, you know, you pick up the high biology textbook and you read the central dogma that within the DNA sequence is your gene encoded. For example, DUX4 shown here with the little arrow showing the start of the gene. So turning on DUX4, by that we mean a process that's referred to as transcription, or making a RNA copy of the DNA sequence shown here. And then the RNA leaves the nucleus, goes out into the cytoplasm of the muscle cell, and is translated into a protein, which would be DUX4 protein in this case. So most small molecule strategies target this first step, trying to prevent transcription or turning on of the gene to produce the RNA. That way you get no DUX4 and, you know, problem solved, but easier said than done. There's been a number of publications on drug classes that can do this, including p38 inhibitors. And of course, you know that the p38 inhibitor, losmapimod, is currently in clinical trials with Fulcrum Therapeutics. Another strategy, again, is targeting the RNA, as Eric again mentioned, with different types of molecules that will cause the degradation or block this RNA function. And Kelly will talk about this in the next talk. And a key here is that these are generally not something you could take with a pill. It have to be injected some sort. And you need a delivery technology to get these things to go to muscle. And that can be either viral or non-viral, for example, an antibody. And finally, as Eric also just mentioned, there is work ongoing to find small molecules that might bind to DUX4 itself and prevent DUX4 from having a toxic function. So Eric actually did another very good job of presenting, you know, the compaction of DNA wrapped around histones in these nucleosomes that are referred to as beads on a string, and then these get packed together in higher order structures, eventually resulting in the chromosomes you see in the cell. Well, one hallmark of FSHD is that you get less compaction and more open chromatin at, in very specific ways. So there are lots of different ways to compact chromatin, and only specific ways are altered in FSHD. Um, so moving on, so in this open DNA, DUX4 is in here somewhere, and what do you need to actually turn it on? So if we just look at the DNA sequence at the top, and again, the gene for DUX4 is on the right. One thing about turning on genes is that you have to have sequences that are regulatory in nature, such as this promoter sequence and enhancer sequence up here. And for all genes, you have to have uh, general transcription factors and specific transcription factors work together in order to tell this gene to be transcribed into RNA. So turning on this gene involves this process we call transcriptional activation. Um, Open and decondensed chromatin facilitates this process, but there's more than that. You need to, actually turn it on with all these transcription factors. And so small molecules could potentially affect both of these processes. And many groups are actually looking into different drug classes that could potentially regulate and decrease the expression of DUX4, decrease the ability to make RNA copies. So the end result of this is that you get epigenetic derepression, so the changes in chromatin to make it more open, and then this variegated expression. And by that, I mean it doesn't happen in every nuclei. What you're looking at here is a collection of FSHD muscle cells in culture that have been induced to join together and form those myotubes. This is part of myogenic differentiation, where you start to express genes like myosin heavy chain here, which is shown in the red. And so you get a long little tube. It's a basic building block of the muscle. It's going to join the muscle myofiber to eventually start contraction. And what you can see in the green here is this cluster of nuclei expressing DUX4, and this is sort of a hallmark. Whereas this myotube next door, you don't see any DUX4 in the nuclei of these, and maybe you see some happening over here. So this is referred to as variegated expression. And this is our cell model of FSHD. So I want to try and put this in the context of what happens in your muscle in vivo. So, if you look at this, we'll call it a myofiber up top, in which left to right, it's the unit of the muscle that will contract, pulling towards the center. When there's an injury, you activate these resident stem cells called satellite cells, which start migrating, and they undergo a couple processes, one of which forms these myoblasts. So myoblasts are cells that proliferate, they grow to large numbers because you want to fill in the muscle void of the injury. And then they undergo fusion in this differentiation step to form a basic myotube, which joins with other myotubes and myofibers and matures until you have replaced the muscle. So I mentioned our cell culture model. It sort of recapitulates this step where you have myoblasts growing on culture. This is a very popular model that many people use. And the reason is that these myoblasts keep growing and dividing so that you always have a population to work with. It turns out that during this process of fusing, certain enzymes like P38 are activated. And that's why we think that inhibiting P38, well, it turns out P38 is activated during normal muscle differentiation. P38 also activates DUX4, and it turns out that DUX4 expression is very sensitive to P38. I'll show a little more of that later. But this is our cell culture model that results in a high level of DUX4 being expressed, even if it's a few nuclei. But one thing the cell culture model can't do is look further down the road when these myotubes mature and join other myofibers in a process that takes a couple weeks. And so we use a mouse xenograft in which we injure a mouse muscle and we actually inject human FSHD myoblasts that we can grow up in large numbers. And as the mouse muscle regenerates, those cues stimulate the human cells to regenerate and become part of the mouse muscle. And that way we can study what happens to them within a muscle in an animal. So this is what it looks like in the lab. We grow up large number of cells. We injure an immunodeficient strain of mice. We injure their muscle and inject the human cells at the same time. Those cells, which were proliferating myoblasts, start differentiating into myotubes and myofibers, and during that process, DUX4 is turned on. So when we use this model, and this was done in my lab, independent of Fulcrum. In fact, at the time, I didn't know Fulcrum was working on this. I tested losmapimod in this xenograft model. And if you look at vehicle here, this is the amount of DUX4 that is expressed when animals are not treated with drug, and that if you use increasing doses of losmapimod, you can suppress the expression of DUX4. So we can express DUX4 by 80 to 90% in this model. So how does that translate to humans? We're not sure how much suppression of DUX4 you need. I'm not sure. That's $1 million question, and can probably, these sort of questions need to be answered in a clinical trial. One thing that I did notice is that with these levels of suppression of DUX4, that the muscle cells did differentiate normal, became part of those mouse muscles, and actually more FSHD cells survived with treatment with losmapimod. So this was sort of the type of data that would get us excited to go ahead and say, maybe we should test this class of drug in humans. So, which brings me back to, this is another picture of DUX4 that's made a little more fancy. It's from this review down here showing a hypothetical patient with one full repeat. There's always a partial repeat at the end with your 4QA allele polymorphism. Sometimes there's a partial repeat upstream, but in this hypothetical patient, as I mentioned before, there's these control sequences often upstream of the gene, and some myogenic cancers have been described, but how do P38 and other factors combine to turn on DUX4 when the chromatin is partially decondensed, which is what we see in FSHD? And that's an area of study in my lab, and other people are looking at this also. But one thing we know P38 does, for example, is it turns on transcription factors, or these proteins that might bind up here and help turn on DUX4. So, P38 is turning on one factor, which then turns on DUX4. And P38 also is known to fine-tune the chromatin structure around genes to enable their expression. So That's an ongoing area of research, and hopefully we will identify new drug targets that way. So I'd just like to sum up that small molecule drugs can turn off DUX4. They do so potentially in many ways by um, um, as I mentioned in the last slide, activating transcription factors and modulating chromatin. And having open chromatin to begin with is one of those necessary things to turn on DUX4. And that's why it doesn't happen in normal cells, because the chromatin is shut all the way. P38 inhibitors and other drugs can suppress DUX4 without inhibiting muscle differentiation in FSHD muscle cells in culture and in our xenograft model. So those are very important when we're trying to find a drug that any drug that inhibits DUX4 doesn't disturb the normal muscle differentiation program. Losmapimod is being tested by Fulcrum, and they've announced that they'll be talking about their results later this month at the FSHD Society International Research Consortium. How these inhibitors suppress DUX4 is currently under investigation, may lead to new drug candidates, and also inhibitors of the DUX4 protein are also being pursued. And I'll stop there. Thank you.